Oh, I was like, what is happening? Why am I not seeing anything?
Recording in progress.
Try this. I'm gonna just turn this down really low. Might as well just turn it off. I'm gonna do it that way. Okay, whatever. Okay, well, it's just not gonna have music. That's fine. Now at least people online can participate. Okay, so we're gonna start. And again, this is going to be the treasure thing. So you're going to want to answer your questions, and then you'll get have to pick the treasure, but be sure you don't pick the trash in order to get your points. And that's the goal of this one. So we'll review chapter 15. It'll be helpful before we get into chapter 16. It's going to be silent in here, and I apologize, but I figure we don't want my voice echoing. Oh, good job, guys. All right. Full screen. All right. Let's see. Okay, see. I noticed something went wrong with you. Chapter 16. I had it pulled up, but it crashed. Sorry. Right. So we zoom. Okay. Okay, got it. Go away. All right. So chapter 16. So we reviewed chapter 15. Do you guys have any questions about any of the questions on the review little quiz thing? Seems pretty straightforward. Good. Because a lot of what we're going to get into with chapter 16 is going to rely on our knowledge or understanding of what was happening in chapter 15. Yes. So cool. Okay. Chapter 16 is dealing with adaptive immunity. Remember, 15 was innate immunity. So that's the immunity that we have just naturally, like recognizing peptidoglycan or seeing LPS, things that are like very general kind of reactions to things. And so that's innate, but now adaptive, we're going to be reacting to very specific things on our pathogens, our targets. Okay. So that's the idea here. First of all, in order for it to be effective, we need immunocompetence. And immunocompetence is just referring to our body's ability to react to a vast number of antigens, essentially. What are antigens or immunogens? Antigens are things that can be seen and identified by the immune system, which we learned, right? So anything you can have, anything foreign, you can have a reaction to with your immune system. Why do we have also this term immunogen up here? An antigen is something that could be reacted to, big picture wise, could be or are reacting to, but once we react to it and have a response to it, it's an immunogen. So that's kind of the difference. It's like once you have seen it and started reacting to it, we call it an immunogen. I'm almost never going to refer to it as that, just so you know. I don't know. I just don't think of it that way. So we're mostly just going to refer to it as antigens. There's going to be a couple slides. The slides call it immunogens. And so that's what, so you know what I'm talking about. All right. So PAMPs versus our immunogens. Okay, here goes verse 1. PAMPs, remember, those are our pathogen-associated molecular patterns, like peptidoglycan and LPS and double-stranded RNA. Very general things that you can say, this is here, this general molecule shouldn't be here. And so we can go ahead and eat this up and digest it, as opposed to your antigens or your immunogens, which are essentially going to be identifying a specific part of a specific pathogen. So as opposed to, I wish I had my fidget spinner here. I wish I have one in here. Because this is, I feel like, a good time for me to, yes, this whole time. Okay, so if, we have bacteria in general, we can see that you have some surface molecule like peptidoglycan that says bacteria to our, phagocytes. Remember, phagocytes are going to be your monocytes, your macrophages, your dendritic cells, your neutrophils, those guys. So those are our main phagocytes, and there are more than that, but those are our main ones. And essentially be eating up all this stuff and digesting it just based on this is bacteria, as opposed to when we have our adaptive immune response, we are reacting to the fidget spinner part of the bacteria. So some molecule on that bacteria, specific to that bacteria, This bacteria has fidget spinner molecule on it. That's going to be our antigen. It's going to get more complex than that. So we need to understand that this is our antigen, but you don't react to the whole fidget spinner. You're going to react to part of the fidget spinner. And that's the epitope. And we're going to get to that. But that's our antigen. Think of it as spike protein on a virus. That's an easy way to look at it. So when you react to the COVID spike protein, is that like when you have that reaction to COVID spike protein, are you also would be reacting potentially at the same exact time to flu spike protein, to any other viral spike protein? No, you're just reacting to COVID spike protein, right? When you get a vaccine, are you getting a vaccine against all viruses ever? No, you're getting it against that one virus because we're looking at that spike protein or whatever it is, okay? How your body sees that spike protein versus how my body sees that spike protein can be dramatically different. Your antibodies against that spike protein are not going to react in the same way as mine do. They don't see the same parts. It's just based on how your genetics designs your antibodies. And that differs and doesn't necessarily transfer from parent to child. This is not something that is necessarily coded by genetics. It's really complex and it's beyond genetics. So that's, it can be transferred to some extent, of course. That's where we get allergies and things like that being able to pass down. But like it ends up being much more complicated than that because we have to be able to make as many antibodies as we do. And that's going to make sense in a minute. Remember, when we think back to your chapter about transcription and translation and what genes are and what they encode, a gene encodes a protein. And every protein that you're going to be making in your cell has to have a gene that encodes for it, except for your antibodies. They're different. So we're going to talk about how that works. And not just antibodies, your T cell receptors too. They work the same way. So keep that in mind. This is how it gets so specific and so diverse, okay? So characteristics of our specific immunity or our adaptive immunity, they're specific. Obviously, we can't say that enough. Really, honestly, we can't say it enough. The response in your adaptive immune system is going to be extremely specific. So specific that your antibody reacting to the COVID spike protein is different than my antibody that my body made against the COVID spike protein. We have diversity. We have at least one cell that can react against any antigen. You might have immune cells that can react. So if this was fidget spinner, your spike protein, whatever it is, fidget spinner, you might have cells that can react to this part of fidget spinner and cells that could react to this other part of fidget spinner in one infection situation. Or you could have fidget spinner on the surface of your virus and also controller on the surface of your virus. And you have some cells reacting to different parts of fidget spinner and other cells that are reacting to different parts of your controller, all in one illness. So when you get sick with COVID, when you get sick with the flu, or when you get your vaccines for these guys, you're not making one antibody. You're making a whole slew of them against different parts of these guys. And so that's what's really going on in the background. So it's not just one reaction going on. So that's why it says you have at least one cell that can react against any antigen. You will never have more than one cell that reacts against one epitope. So what I'm saying is you might have one cell that recognizes this part of fidget spinner and another cell that recognizes that part of the fidget spinner. These are different epitopes on the same antigen. So there's two different cells getting involved here. That's what it's talking about when you have reacting to an antigen with more than one cell. But you could potentially have cells that could react to any possible imaginable antigen in the entire universe based on what we understand about chemistry, unless there's other chemistry out there in the universe that we haven't discovered, possible. But if there isn't, then your body, just your body, can react to any antigen possible in the whole universe. That's 8.6 billion different possible antibodies we're talking about. 8.6 billion. You have 20,000 genes in your body. So what did I just say about proteins? That you need a gene to make a protein. Antibodies are proteins. So your cells are about to get real dirty with your DNA. And I'm not kidding about that. These guys exist to basically mess up that DNA code in a way to get as much diversity as possible out of it. And there's a whole system in place for that you guys luckily don't need to know super details of. We'll touch on how it works a little bit. But that diversity is basically why you're alive right now, honestly, of all the things that we see in life. And the reason that humanity can survive for as long as it can, yes, it's awareness of like what we should be eating and what we shouldn't. And, aseptic technique has a big part of that, washing your hands and all of this. But the things that you aren't being protected from there, we have vaccines and we have our immune system being strong enough to deal with all these diverse things that we're still going to be facing up into, 100 years old. So it's very impressive. All right, we have inducibility. So we have all these 8.5 billion, whatever different antigens, and each one we have a cell for, right? We have to only induce the one that recognizes fidget spinner, right? If we're having an infection, we don't want to activate any of the ones that are associated with other infections because you're wasting your body's resources at that point. So you have to have a way of only inducing the response that you need and not activating every cell in this whole situation. When you have an innate response, there are signals that are sent out to basically call everybody in. There's not discrepancy here. We're sending out, come and help and eat everything. When we get the adaptive immune response, you do not want to call every bomber in your arsenal into that area. You only want to call the ones that you need into that area because it gets real nasty really fast if you overstimulate your immune response. That is what we get when we talk about the cytokine storm and the effects of the cytokine storm. We'll talk about that too. But anyways, inducible, it's clonality. So if I'm telling you there's one cell in your whole entire body right now that can recognize this part of fidget spinner right here that I'm holding on to, there's one cell that can bind to that in your body right now. We will multiply. We recognize that guy gets activated. We get the signals that we need to activate him. And that guy will multiply out. Of course, we're only activating him. And then he will multiply out. He's not going to stay one cell. He's going to say, I've been waiting my whole life for this. Finally, I've seen this, and will multiply himself out and then turn into all the specific types of cells that we need to start fighting the good fight. We have tolerance. We're supposed to, while we're going through our maturation processes in the bone marrow or the thymus, depending on if you're a B cell or a T cell, respectively, supposed to be getting rid of any of the cells that could identify self. This is part of what you do when you're just growing up. You should be getting rid of them pretty early on in development, actually. So supposedly, you're supposed to be getting rid of all of them. Now, unfortunately, there are some of these cells that recognize, part of something, like part of fidget spinner, for example, but you might have antigens on your cells that don't stimulate self. Like they don't call out as saying, I am self molecule reacting as strongly as it needs to in order to get rid of these guys. But it is strong enough that whenever they're activated later on, they're going to attack yourself. So there's gray area there that we can't always catch early on in development, or for some reason, we didn't catch it early enough in the maturation process to get rid of it. We didn't have the right signals or the right interactions between, molecules and whatever else that you need in order to signal to get rid of somebody who is reacting with self. We also are supposed to have strong responses in the adaptive immune response. If there is an illness going on in your body and you have a cell that reacts really strongly to this part of fidget spinner and one that like kind of associates with the other part of fidget spinner, but it's not like super attached to it, your genes just didn't quite align in a way that it's a super strong response, your body will just kill that cell. It doesn't want to mess with that. We want strong, definitive responses. We aren't messing around with anything that aligns with self or that gives us weak interactions. That's a waste of our time. So that's how that's going to start going. So now you can see how if you can start out reacting to 8.6 billion different antigens, possibly, that includes self and includes that weak crap too. So you can see how you're already starting to whittle down on your, cell repertoire because of that. And then, of course, the biggest part of adaptive immunity, the most important aspect of it would probably be the memory aspect. We're going to have a strong response, and that's great for us when we're having the active infection, but it's so much more beneficial that we can remember that we saw this part of fidget spinner. And that when we see it again, we don't have to go through all of the whole process all over again. We can just wake up the guy that flew that bomber and say, you remember this? And he says, yes, and just starts, throwing bombs at the guy. We don't have to wait for the other crap. So that's the importance of memory. So it says rapid mobilization of the lymphocytes that are programmed to recall the blah, blah, blah. So seeing that thing again, having cells that can live for almost your whole lifetime. It just depends on the cell and the antigen we're talking about. Sometimes years, sometimes decades, and sometimes for an entire lifetime. Those cells can just hang out and just be waiting and waiting to see that thing again. But they've already been triggered and they've already been activated. They just are basically centuries now. Okay, so this is the whole idea of how we have, development going on with our lymphocytes. As far as what we need to know from this chart, essentially, is just that B cells are going to be making antibodies, and the T cells are going to be involved in the cell-mediated immunity. Okay, so why isn't B cell-mediated immunity? Because the antibodies are the ones that are determining what's happening in the immune response on the B cell side. They're the ones that are doing things. The B cell's just making the antibody. They're not doing crap. They're just chilling, pooping out antibody. They're not doing anything else. So they are not actually that active in this. Okay. The antibodies are the ones doing things. And are they even doing things? I would argue no also on that, but we'll get to that. The T cells, though, easily, so many people think of how important antibodies are, how important B cells are. T cells, easily the most important parts of your adaptive immune system, hands down. Because without the T cells, you can't activate the B cells. So the T cells, our T helper cells specifically, are our captains in all of this response. You do not move without the captains go ahead. The B cells do not make antibody without the captains go ahead. The T cytotoxic T cells are tanks that are going to go out there and kill. They do not act without the T helper cell go ahead, the captain. The T helper cell is the captain that's going to be controlling the responses of literally all of the adaptive immune response. So when we think about that, the T helper cells being so important in controlling the activation of B and T cells in general, our T helper cells are the ones that are going to be infected by HIV. HIV infects those cells preferentially. And so if you are infected with HIV and you are killing off your captains, you cannot activate your B cells and you cannot activate the tanks of the T cells, the killer T cells. So you see now what you see with AIDS, as we have a severe decrease in your helper T cells. That's all they look at is your helper T cell counts, your CD4 positive T cell counts. The only time that's all they look at. And you see people that are getting cancer. You're seeing people that are getting infections from things that no otherwise healthy person should be seeing. Think about that for a minute, because what that means is those things are things that your body are seeing right now, every day, day in and day out, including the cancer. And your immune system's taking care of it because you have your captains to tell the other cells what to do. And I'm dead freaking serious when I say even the cancer. These cells are involved in maintaining control of cancers as well. Now, these aren't super common. I'm not talking about like, acute lymphoblastic leukemia or breast cancer. I'm not talking necessarily about that. I'm talking about things like Burkitt's lymphoma and Kaposi's sarcoma, which might not sound like anything obvious to you, but if I take away your immune system, it'd be pretty freaking obvious to you pretty fast because your cells are keeping that cancer down. Burkitt's lymphoma is a cancer that you get when you have mono and those cells aren't being kept in check by your immune system. So you get cancer from mono because mono lives in the cells in your lymph nodes. It's a herpes virus, by the way. So it's going to incorporate itself in your DNA. It's permanent and it's forever. So even if you had it as a kid, if you get cancer, if you get HIV later in life, you could start having cancer come out from having had mono when you were 5. And that's how this goes. That's what Burkitt's lymphoma is. Your captains of the whole thing, which is our T helper cells, If they aren't working, we can't keep that in control. That starts to put it into perspective exactly what these guys are doing for you, not just when you have your poopy old little cold or COVID, but all of the time, every day, day in and day out, always. It's the reason why you don't die just from going outside and seeing the bacteria out there. It's pretty interesting stuff. If this is your life-saving body system here. If you didn't have immunity, you'd be boy in the bubble. You would die. That's just the end of it. So, all right, let's think about this. So, and we'll talk about that stuff like immunodeficiencies and stuff in chapter 17. That's coming too. So we will be discussing AIDS, and we will be discussing like SCID and things like that in chapter 17. So we'll talk about that more. But now that we're starting to understand the importance of this, we can get the gravity of exactly how severe these conditions can be. I just remembered that I didn't hand out the sign-in sheet, but that's fine. So this is just showing us about how we have our B cells that are going to be developing in your bone marrow and then moving into your lymphatic tissue. Your T cells are going to be starting out in the bone marrow, and then they're going to be moving into your, usually in circulation, but they'll hang out in the lymphatic tissue as well. But they are all over the place, okay? And they better be, because they're the ones that are going to be associated with making sure all the other responses are going down. Okay, so this chart is nuts, and I absolutely despise how they set it up in these slides. Obviously, these slides were pre-made by McGraw-Hill, and I just try to edit them for my use. But this, I just hate it, because yes, I have to have half of it up here and half of it down there so you can read it. But honestly, I'm not going over this right now, because it's not going to make a whole lot of sense until after we go through all the cells. And then you guys, I recommend going back through this after we've talked about the whole immune response and trying to see if you can figure it out, and even redrawing it so it's not on 2 slides, okay? So whatever you need. This is also a thing that we will kind of be going over whenever we do Friday, tomorrow, I guess it's Friday. When I am done giving our weekly thing about things that people don't do well on, I'm going to, instead of, like I said, instead of going over Dr. Morford's thing, I'm going to go over the immune response. And so it's not just going to be this, it's going to be the innate response that leads up to this and all of this. So I'll be walking us through all of that. So if you guys want to wait to see that, to redraw it, that's great. But at some point, I would definitely re-look at this and make sure that it makes sense, or that you can make it make sense to you. Because this is just describing your entire B cell and T cell response and what's going to happen downstream of that and why it's important. But we're going to go through these step by step. That's why I'm not talking about this right now, right here. First, we're going to start with the major histocompatibility complex. It's called the MHC. It's also known as the HLA, or human leukocyte antigen. I used to work at Integris in the transplant lab, transplant testing lab, whatever you want to call it, but we called it the HLA lab because what we were doing was testing people's HLA. And that is because this is our molecule that if your macrophages and your other antigen presenting cells, macrophages, monocytes, B cells, whatever, have gone and cleaned up that whole mess where that the neutrophils left behind and picked up what they liked in that area. And they're like, interesting. Does anybody recognize this, right? Remember how we were saying that those cells were eventually going to be going around saying, does anybody recognize this? I have a fidget spinner here. Does anybody recognize this? My hand that's holding up this fidget spinner right now, my hand is the MHC. Okay, so that is the major histocompatibility complex. It's called histocompatibility because if this was just floating around, if a fidget spinner, just for those of you who are online, so we have quite a few of you now today, if this fidget spinner were just floating around in your body, your T cell don't give a crap about that. It's not going to do anything with that. And then your B cell can't do anything with it because your T cell has to activate it first. So why does that matter so much? If the MHC is holding the fidget spinner, it says, I am a self cell. I found this crap. And self cell says this is crap. Let me show you. Do you also recognize this as crap? And then the T cell says, yes, indeed, I do recognize that as crap. And that's going to be our T helper cell recognizing fidget spinner in hand or antigen in MHC. If it doesn't come in that combo, it doesn't care. Why? Why would that be so important? Because we have to have checkpoints in this. When I am making this comparison of your B cells, B for bomber, and your T cells, T for tanks, if we're bringing in tanks and bombers into the fight, you don't want to start killing your own guys. And you don't want to start killing everything in that whole area. For example, in your gut, do you have bacteria in your gut that have peptidoglycan? Yeah. Those are technically, otherwise would be seen as foreign. But our body had to train itself not to attack the guys that are in your gut, and specifically because they're in your gut. We have a lot of very intricate interactions going on here. We can't afford to just have willy-nilly reactions. So having it in the MHC is part of that checkpoint. It is how you're going to interact your antigen with your T cells receptor. Okay, so if I am macrophage, and generally this is how I'm going to be showing it to you guys, but if I am macrophage and I picked up this crap from the neutrophils at the infection site and I found fidget spinner and I'm holding it out on my MHC, now the T helper cell, that one T helper cell that recognizes that part of fidget spinner, that antigen, that epitope of the antigen, who has literally just been sitting there in the background saying, I have waited my whole life for this moment, literally been waiting for this moment, finally comes out and says, I do recognize that. Y'all in the background waiting on your antigens can suck it, it's my turn. And then comes up and then recognizes its epitope, and we send signals to each other. I send my signals to the captain, and he sends his signals to me. And as a macrophage, I'm going to go back out to the fight and keep eating and sending signals and activating other cells potentially and whatever. The T helper cell, though, now that we've activated him, can start the entire activation of the rest of the adaptive immune response. That interaction has to happen, whether you're going down the B cell route or the T cell route. So that's A number one. Okay, so we have different kinds of major histocompatibility complexes. We have two kinds that you guys need to care about. There's more than that. You're welcome. Okay, two that you need to worry about. This is how we recognize self versus non-self and foreign, making sure we're reacting to foreign appropriately. We have a checkpoint there. Okay, so we have class 1 and we have class 2. And I know here it has class 1 with a number 1. Usually when we write MHC1 and MHC2, it's going to be with Roman numerals. Okay, so that's kind of what the 1 down there is depicting. So MHC1 versus MHC2. Now, if I am a macrophage, me, which is what my little example is, right? And I go to the site of infection where neutrophils have just, pooped out all over the place, and I pick up their waste and I see fidget spinner and I take it inside of me and I'm displaying it now on my MHC. That MHC that is holding it up at that point in that particular situation is MHC class 2. Because, and this is it, MHC class 1 is present on every cell on your body except for red blood cells, every nucleated cell in your body. Your red blood cells do not have a nucleus, they don't have DNA. So they don't need it. All they have to do is carry around oxygen and crap like that. Who cares? So your class 1 molecules are going to be, and the reason that we don't care about it with red blood cells is going to make sense in a minute. Class 1 molecules are going to display fidget spinners that came from inside of the cell. Whereas later on, I'll say in another slide, the call came from inside of the house, right? So what's going on is you have a virus in your cells actively multiplying inside of you. Something's wrong inside of your cell. That's MHC 1. 1 looks more like an I to me, especially if it's Roman numeral. I being inside. It came from inside. It's presented on MHC 1. Okay. So that's going to be viruses. It's also going to be any other abnormal thing that shouldn't be around in the cell. Like if you have cancer, you will have proteins and things that are being formed at wrong times, wrong points in the cycle, whatever it is like that, that can also be presented on this. So we can attack cancer cells this way too, which is where that protection comes from that we were talking about previously. So any cell in your body can do this because any cell in your body can have a viral infection, except for your red blood cells, which don't have a nucleus. So can viruses multiply without your nucleus and your everything related to your nucleus? Absolutely not, right? They need all of your machinery. If you don't have the machinery, what's the use? So we don't need it on our red blood cells. The other cells do. So there we are with class 1. The call came from inside the house. That's what's happening. So it's like if I was an infected cell in your lung whenever you have COVID or something, okay, and I have a virus growing inside of me, it is like if you were an alien, okay, an alien, the alien grows inside of your chest. You get the little face hugger guy, and he implants the embryo, and then it bursts out of your chest. It's my favorite sci-fi horror movie, by the way. So if you guys haven't been in my office, I actually have a little statue of the alien in my office. I'm like obsessed with it. So anyways, the alien is inside of you, and you know it's inside of you, and you're like, oh my God, I have this alien inside of me. What are we going to do? Well, like in the movies, if you were in the movies and you watched all the other movies, then you would know at some point the people who get the aliens in them when they know are like, just kill me, because they know that what's going to happen, right? That is exactly what's happening with MHC class 1. Your cells are saying, I've got the alien in me, kill me. And that is what will happen. So we'll get to that, but that's MHC1 and the importance of MHC1 and why it is on all of the cells. MHC2 is not. MHC2 is only on your antigen presenting cells. The antigen presenting cells, there's only 3 that you guys need to know of. The macrophages, the dendritic cells, and the B cells. So we know macrophages and dendritic cells came from monocytes. So you could put monocytes and their derivatives again here if you want to. But really, if they're already reacting to this, they're probably differentiated into tissues. So that's why we say macrophages, dendritic cells. And then B cells. And this is a new one, right? This is not one of our phagocytes that we were talking about earlier. But we consider B cells to be part of your adaptive immune response. So how are they presenting antigen as our initial presenters in reaction to seeing foreign things in our body? So what's happening is our B cells, who also recognize fidget spinner, it may even be the same freaking epitope as what your T cells would recognize, right? So yeah, my macrophage, I might pick things up outside of my cell. So like we said, MHC2 is stuff from outside of the cell. So it has to be. If we said MHC1 is from inside, MHC2 has to be from outside. A lot of your bacteria are outside of your cell. You can also find viruses outside of your cell too, though, right? They're not always inside of your cells. So we're starting to get into this picture. Okay, so we have the MHC1 reacting to the inside stuff. We have MHC2 reacting to the outside stuff. So more likely than not, you're probably having both involved in an infection. We'll have signals. Of course, cytokines, we're back to that again. We'll have signals from the cells telling each other what we need more than the other. Do we need more cell response or do we need more antibody response? And we'll have signals telling us what to do there. And you don't have to worry about what those signals are. We just know they're called cytokines, okay? But that'll help drive us if we need more of one response or the other. Because antibody is going to be way more effective for things outside of the cell. Antibody doesn't get into your cells. It only stays outside. So it's not very effective against things that are already in your cells. For that situation, we have to kill the cells. And if I'm telling you antibody doesn't do that, what does it mean? What cell does do that? A T cell does that. Okay. So MHC2 is for things that are outside of the body. B cells are one of our antigen presenting cells, in addition to our obvious macrophages and dendritic cells, which are just like macrophages, but in the skin. And the B cells, what they do is they're nice and happy. Remember how I told you when they mature, they just hang out in the lymphoid tissue, whereas the T cells are kind of just all over the place. Well, the B cells literally just chill, reading their books or whatever in the lymph nodes at those checkpoints as everybody is coming out of that infection area through the lymph. Before we go back into circulation, they get filtered through those lymphoid tissues. So they're going to pass through all of these B cells that are just sitting there waiting and looking around, waiting for the fidget spinner that they recognize. And just like with the T cells, if they see one that they recognize, they're going to grab onto it. But they can't do anything at that point. They have some minor things that they can do that are involved in the innate response, but they can't make antibody, which is their main purpose. They can't make antibody without the captain's go ahead. So they have to find B cell chilling at the checkpoint. We found a bad guy. Okay, we'll take part of this bad guy, take his picture, go show it to the captain. When you find the captain that matches your picture, your fidget spinner, now he can say, yep, let's go. It's go time. Make your antibody and I'm going to get the tanks. That's what's going to happen. But you have to have the go ahead from the captain. So B cells are there. They're doing their thing. They're not phagocytes in the traditional sense, though, but they are antigen presenting because they can pick that stuff up during that filtering process through your lymph nodes. Really cool to think about how that might work because you have your macrophages and your dendritic cells in your tissues, but your B cells are there filtering out your lymph all the time through your lymph nodes and everything, waiting for the right opportunity. So this is looking at the receptors on the surface of our B cells and our T cells. That should look pretty familiar to you. On the left, these are antibodies, right? That's what we learned about what an antibody would look like when we talk about it in lab with ELISA and whatever. I'm terrible at drawing it, so it's a better depiction than what I draw, but yes, but it's stuck to the surface of a cell. So this is immunoglobulin. An immunoglobulin can be attached to a cell or it can be secreted. When it's attached, of course, it's our B cell receptor. When it is secreted, it's the same dang thing. It's just now like in the, what I say, soluble, that's what we'll say. It's just floating around, it's being secreted. And that's antibody when that's happening, okay? But it's the same molecule, it's just whether it's attached as a receptor or if it's being secreted. The T cells don't do it that way. They draw it so differently in the picture, but they have essentially the same kind of concept on how their structure is, but they never secrete. They're just always attached to the surface. They also come along with this other molecule, this accessory molecule of CD4 or CD8, depending on what your cell type is. And we'll get into that more, don't worry. All right, we already talked about antigen presenting cells, dendritic cells, macrophages, and B cells. Whenever you finally get activated as a B cell or a T cell, whatever kind of T cell or B cell you are, you're going to proliferate first. Then you're going to differentiate into your different types. There are 3 types for every one of these guys. If you are a B cell, there's 3 types. If there's a T helper cell, there's more than 3 types, okay? There's more than 3 for the T helper cell, but there's 3 main types, okay? And then 3 categories, I will say. And then the cytotoxic T cells will have 3 main categories as well. And those main categories are effector cells, EFFECTOR, effector, they're having an effect. They're doing the work, right? They're making the antibody, or they are the captain activating other people, or they are the tanks going out and killing cells. Or you could be regulatory cells. I feel like that speaks for itself. Regulatory cells are just going making sure you're not overreacting. Or you can be memory cells. So any of the B's and T's they will become those 3 every time that they're stimulated. So that's the differentiation part of it. So, but you have to multiply out first, and then those will become the subsets. All right, so our T cells, how are they going to be responding to these things? These are we say they're tanks, but they're technically our tanks and our captains. So we can kind of divide it out that way. But we have our T helper cells, and of course the T regulatory cells that come from both sides, and the cytotoxic T cells. Now, the cytotoxic, we talk about T helper cells, they're the captains. The cytotoxic T cells, these are the ones I keep calling the tanks. The ones that are going to go out and assassinate the infected cells. If you're sitting there with a virus in you and you're saying, I've got the alien about to burst out of my chest, help, help, help. These guys are the ones, the cytotoxic T cells, that will go out and kill infected self cells. So you can start to see now that the cytotoxic T cell response must be regulated and heavily, because you don't want them going out and killing self cells if they're not infected. You need to have checkpoints in place. So this is even way more important, I feel like, having those checkpoints in place than you need checkpoints in place for antibody, because you really don't want to be killing self cells. I'm telling you that these are the cells, these cytotoxic T cells, these are the reason why you don't have Burkitt's lymphoma and Kaposi's sarcoma right now, because they're killing those cells. They're keeping cancer at bay. That is some heavy shit. Like that is like, you don't want to accidentally do that. This is so serious. These are the guys that whenever I'm sitting here saying, help, I'm infected, I've got the alien in me, and I'm holding this out on my MHC1, my fidget spinner, right? Everybody has MHC1 on their cells. Your MHC1, and the whole reason that HLA, which is the same thing as MHC, the HLA lab only focuses on MHC1, HLA1, because all of your cells have that. And all of my cells have that, right? To be able to say I'm infected. But your HLA molecules look different than mine. Your genes have made it so that they can see that as self. But what your body sees as self is for you to see as self. You put your kidney in me, I'm going to see your MHC molecules that are just chilling. They're not screaming, hey, I have an alien in me. Your kidney cells aren't doing that. So they're just, just chilling, like with their, hand doing normal stuff. But my body sees it as, help, I'm infected, because it looks weird to me. It looks like MHC, but weird. So my body will attack it and kill it. It will destroy entire organs within an hour or two if you don't match, and there's already predisposition for that attack. You can see it happening during surgery. It can be that quick if they've already seen that antigen. That is devastatingly dangerous. So we have to have checkpoints there. That is why they do so much testing when you have to get an organ transplant. Yes, because finding somebody that matches you adequately can take a very long time. So we're looking at every possibility of every mutation ever in HLA or MHC and making sure, okay, well, we've looked in the past, you have this mutation and this person's a little bit different, but we have never seen any cross reaction with that. Maybe that'll be okay if we can't find an exact match. Or these mutations here, where we see that happen, it always ends badly. The likelihood of this person rejecting is high. And even when you do that, we still see, if we match it up perfectly and do all the testing, we still see people rejecting, which is what happened. I think I told you the story about my friend that needed the kidney. I didn't tell you guys about that. When I worked at HLA, one of my friends came through. She has type 1 diabetes, and she needed a kidney transplant. And I did all of her testing. Of course, I couldn't say that because of HIPAA and all that. But I did all the testing for it, and everything was clear. Everything was good. And within about a year of her getting her new kidney, she rejected it. And there's just shit you can't test for. And that's just how it goes. Like, it's not always all about HLA. Sometimes it's about, no. She had to keep going. She took her medication for as long as she could keep up with keeping it like at bay, and was having to have dialysis and eventually had to lose that kidney. I think she's going to get another one, but that's how it goes. It's just kind of like, cross your fingers.
I just asked my son how her
Yeah.
Lots of things.
There's a lot of testing. A lot. And I think people don't appreciate how much testing goes into it. And it's even trickier.
Sorry, there was something that, because he was an infant, and they said as long as you're under 2, like you can get different things for that 2, like your antibodies form.
Yeah, because their immune system is still adjusting. So they could still potentially react to a newer kidney or something, or heart or whatever, and start training their body to see it as self. So they have a better chance than an adult would. So the older you get, the worse your chances, pretty much. Yeah. But yeah, that has a lot to do with your adapting responses early on in childhood. But yes, so whatever. So cytotoxic T cells, they're the ones involved in all that. So we aren't worried about antibodies when we do transplants. We aren't worried about anything else. We're worried about cytotoxic T cells. And yeah, we're going to talk about that in chapter 17 too, in more detail. And I deleted some stuff at the bottom of these. So if I deleted it on these slides, you don't need to know it. All right, B cells, essentially what they're going to do is be activated. Of course, they're going to have their memory and their regulatory cells, but they also have plasma cells. That's their effector cells. Plasma cells are the cells that make the antibody. Once you're an activated B cell, if you're making antibody, you're a plasma cell. That antibody that you make will respond to that exact same antigen that you were initially stimulated by, not anything different. All right, so we talked about these markers on the surface of cells. CD3 is just something that helps with self-recognition. You don't really need to know about it otherwise outside of that. We're not ever going to bring it up again. CD4 and CD8 are our markers that help us determine whether it is a helper cell or cytotoxic cell. So cyto, of course, being cell, toxic being toxic. And these CD4 molecules, so we had our receptor. Remember when you're a T cell and you're looking around for these guys, holding stuff up, saying help or whatever it is, or I found this from the neutrophils, whatever it is, those MHC with the antigen, the T cell receptor is what's going to come and interact with that and say, I also recognize this. Cool, I also see the fidget spinner. Great. But these CD4 or CD8 markers are going to help solidify that connection and saying yes, and help like activate basically the response to that appropriately. So just a marker. Don't need to worry about anything else beyond that. It's just some crap on the surface of the cell, but it helps us identify the difference between the helper cells and the cytotoxic cells. Immunologically speaking, it's how we would divide them up if you had like somebody's sample, whatever. So and a lot of times I'll just refer to something as CD4 positive or CD8 positive T cells. CD4, the 4 to me looks more like an H than the 8 does. Okay, so that's your options. It can be a 4 or it can be an 8, and the 4 looks more like an H to me. So that's my T helper. Okay, that leaves the 8 with the cytotoxic. So that's how I remember the difference. CD4 will only be interacting with MHC2 molecules. That means that those captains, which are T helper cells, have to interact with one of those antigen presenting cells. That's our B cells potentially, or those could be our macrophages or our dendritic cells. So picking up the crap from the area or filtering it through the lymph node, we picked up this extra stuff. And now we got it from outside of us. We have to present it on the outside, on our MHC2, saying we found this outside of us. CD4 positive T helper cells, they will be the ones that can interact with that. And that's going to stimulate their action, their activity. If you have something being presented on MHC1, you can't activate the T helper cells with that. You can't. So if you're sitting there screaming, I have an alien in me, the only thing you can interact with are their cytotoxic T cells. And the only time that those guys can come and kill you when you have that alien in you is if they've been stimulated by the CD4 guys, and they had to be stimulated by MHC2 from outside of the cell. So you have to have all of these steps going in place in order to activate that. You can't be killed with the alien in you until your cytotoxic cell gets activated by the captain. And they couldn't get activated until they saw it picked up from the outside. Because again, if we have a virus, then you probably are having a virus that's also outside of you somewhere. So it should work out. Anyways, so that's part of the whole checkpoint thing. T cells, again, they prefer to circulate, but they can be found in lymphoid tissues. B cells are always going to be found chilling in the lymphoid tissues. They don't need to be circulating because their antibodies are going to do the circulating once they get activated. All right, so here is how we get all that variability in our antibodies whenever we are creating our new genes and all this and reassorting and all that. You have all of these little parts of genes that are associated with that tip of that antibody, the Y, right? And that tip of that antibody at the end of that Y is called our variable region. Because, yeah, guess what? It's variable. It can be changed up. And we can reassort those gene combos and add in nucleotides or take out nucleotides or whatever to get as much combination as we can possibly get. So this is all happening in those cells during their development in like the bone marrow. And so we have that genetic reassortment happening after you've been conceived. So this variability won't be the same from your parents at all, because we have so much variation that can potentially happen. So that's going to be the variable region. And then we have the bottom of that Y. Think of it almost as like a handle, okay? Like if you're holding on to that antibody, you'd hold it at that bottom. And that is our constant region. That constant region is what determines what that antibody can do for the body. Okay. Again, those are called immunoglobulins. And once we start secreting them, we will call them antibody. We have that variable and then the constant. And then that whole long piece that goes through the middle is called the heavy chain, and the light chain, which is the pink that I have out there on the outside. That's just what we call those parts. Okay. More important to me that you understand about the variable region versus the constant region, but that's the light chain and the heavy chain. They're held together by bonds. All right, the T cell receptor is pretty similarly set up. We have a lot of variation in that works the same way, the gene reassortment. It's just different genes. But it's just always going to be in the membrane and never secreted. So then what happens is you have clonal selection. You activate that one that only recognizes that one part of your fidget spinner. It multiplies out. And then it's going to go and all those multiples become different things like the effector cells or the memory cells or the regulator cells and do their jobs. But you only activate that one. But before that, when we had our reassortment of our genes, we had to go through clonal deletion. And that is when we reassorted our genes, we have some of them that are going to react with self. That's just how it goes. So during development, your body should be getting rid of the anti-self clones. That's clonal deletion. That's what that term is. Removing anti-self. And then all you have left should be the cells that can react with appropriate antigens. All right, so immune tolerance is where immune tolerance saying you tolerate self, and immunocompetence being you react to, all these different possible things. So you have to have kind of both going on, not reacting to self. You achieve that through clonal deletion. All right, antigens, again, we're presenting them in order to activate all of this, and it has to be presented by MHC for the very, very, very first initial, response of all of this. It has to be presented to the T helper cell on the MHC2. It just has to, because that's what's going to trigger everybody else downstream. If we can react to it, we call it an immunogen, right? Like we've said previously. In order to be a good antigen, some things are better at being antigens than others. So good immunogens are going to be things that usually it's proteins or carbohydrates, and larger usually is going to be better. Things that are like, don't have a lot of shape to them, like these chains. If they're just a straight out chain and they don't have a lot of shape to them, then they're not usually going to be very good for eliciting an immune response because they don't have a lot of stuff to be recognized, if you will. So generally larger is better. And we like more like complexity to the shape and chemical composition. That's going to be a better antigen. Again, an epitope is the part of the antigen you're actually associating with as an antibody or as a receptor. So it's the part where so that part of the fidget spinner that we're binding on to. If the fidget spinner is the antigen, that portion of the fidget spinner is going to be the epitope. Haptens are antigens that are too small for us to react to by themselves. Toxins usually fall into this category. So tetanus toxin, pertussis toxin, diphtheria toxin, if this doesn't sound familiar to you, the TDP or the DTaP vaccines. So those are all toxins and they're all too small for us to react to. So we've put them together to make them bigger so we can react to them better. So we can essentially do that. If you just wanted to react to one of them, you could put them onto a carrier molecule. So that's what this is showing down here. This is our hapten carrier situation. Too small at first to react to. We add it onto something to make it bigger. Now there's probably going to be an antibody that can react to the hapten portion. Alloantigens, this is just like we were talking about with compatibility and organ transplants. These are things that are found on everybody's cells. We all have MHC, for example, but yours look different than mine. So those are alloantigens. Another good example, of course, that I always forget, which is weird since I worked at OBI, but is the blood group stuff. So I'm A positive, so I cannot receive B or AB. If it has B in it, ain't happening. We'll talk about that in chapter 17 as well. My dad is B. My dad's B positive. My sister's AB positive, but I feel like it's like the optimal. If you're going to be a person that needs blood, the optimal to be is AB positive because you can get any blood type. It's the universal recipient. Universal donor is O neg, but also universal donor being O negative, you can only receive O negative. And it's not the most common blood type at all. So that can be a problem. Because of course, if you go to the hospital and you're injured and they don't know what your blood type is, what blood type are they going to give you? O negative. And so what are they going to run out of fastest? O negative. So yeah, so it can be a problem if there's mass injuries. Anyway, super antigens are things that elicit an overreactive immune response. I don't know what else to say about that, but the things like LPS we talked about in the innate response that can trigger, fever, or it can even trigger, shock essentially. And this is what's happening here with our bacterial toxins like endotoxin. They're going to stimulate the immune response so heavily that we just don't keep it in check with our regulatory cells. They're just not being, they can't keep up with it. And that's cytokine storm. So that's too many signals being sent to tell about like what's going wrong. So that leads to toxic shock syndrome. So antigen processing and presentation, we already said it's macrophages, dendritic cells, and B cells as our antigen presenting cells. And we're not going to go through these because we're running out of time, but there are questions here. What is the difference between PAMPs and antigens? Remember, antigens are very specific to a specific pathogen and a part of a pathogen, whereas PAMPs are going to be generalized for large groups. T cells, complete maturation in the thymus. MHC is associated with pathogens that are found. Which one is associated with outside of the cell? That's going to be MHC2. Remember, MHC1 is inside. What are the major antigen presenting cells? I would have, if I'd have you guys answer the question, because I never remember to save time. Is going to be the macrophages, dendritic cells, and the B cells. And you guys could have named just one. What is the CD marker for cytotoxic T cells? So that's going to be CD8. It's either CD4 or CD8, right? So here's how antigen presenting cells are going to do their job. Essentially, they're going to eat up a microbe, which we see in the upper left. They're going to put it in their little phagosome, right? Break it down in their phagolysosome by mixing it with the lysosome and get its little parts and pop it onto that MHC and sit out there saying, hey, does anybody recognize this fidget spinner? Then what happens is we get a T helper cell in this image down below. Get that T helper cell here. And it will interact with that MHC, that brown part with the pink. Its little green squiggly guy reaches up and recognizes it. That's its T cell receptor. Okay, so that's where we have that variability. It has its own variable region. It's not an antibody, but it has a similar makeup. And because it's a T helper cell, it has CD4 that's also going to help recognize this interaction and affirm it. Now, great. So we've seen it and we're happy and we're affirming that we have this interaction. Now we start making signaling molecules like IL-2, interferon gamma, and IL-4. Interleukin is what IL is. So those are just cytokines. Okay. So depending on what cytokines are made, we can skew our response one way or another throughout the whole process. But ultimately from T helper cells, you get this whole mess of BS. Okay, you get your memory cell, you get your Th1, your Th2, and you get your regulator, and I don't care about the other ones. Okay, so we have our effector cells, which we happen to have two of them here, Th1 and Th2. We have our memory cells and we have our regulatory cells. Just like I told you, there will always be three categories. That's all we care about is those ones. All right, so we are always going to get Th1 and we're always going to get Th2 in this response. And that's important because think of Th1 and Th2 being T helper. They have what? They have the MHC2 response. So their receptor recognizes MHC2. But now with Th1 and Th2 helper cells, these are what's going to essentially skew our response to be antibody or cell mediated. Th1 are helper cells. They're still helpers, CD4 helper cells that will be able to go out and interact with our cytotoxic T cells and stimulate them. So cytotoxic T cells, their captains are the Th1s. Remember it because MHC1 that interacts with those cytotoxic T cells is a 1. So cytotoxic T cells also need help from another 1. So it's Th1. Th2 are going to be the ones that stimulate B cells. So I consider that to be more of the outside stuff. So like the MHC2 related stuff. So the 2s goes with the 2s and the 1s go with the 1s. And those responses you can have as the 1s being related more to the inside stuff and the 2s being related to the outside stuff. So we can keep it in those kind of groups, okay? All righty, cool. Then we have stimulation. So our TH1 that we finally activated in that previous slide, right? I said they're going to help out with the TCD8 positive ones. They're going to give signals along with whatever cell also is holding out that help me MHC1 BS. So MHC1 with a CD8 cell, it's getting the go-ahead from its captain, from the TH1 cell, and it can itself make memory cells and regulatory cells, but its effector cell is the cytotoxic T cell. And what it will do is go out and find even more cells saying, help me, I'm infected. Help me, I have the alien. Please kill me. And go and do that. And it makes these things called perforins and granzymes. The perforins perforate or create holes. And the granzymes are enzymes that are going to be degrading the insides of those cells. And eventually will cause the cells to die via apoptosis, which is essentially cells unaliving themselves, okay? So I had another student that was like, you sound like you watch a lot of YouTube gamer videos. And I said, that is accurate. Okay. Right, so with the B cell, we're going to need that secondary signal from our T helper cell down there. It's kind of in the middle-ish where we have B cell growth factors, and then interleukins are signals, signaling back and forth with one another, activating one another. Because again, we might be naive. Your B cell can be an antigen presenting cell. So if it is, we have to have signals going back and forth, activating both the helper cell and the B cell. Then that B cell can go through its BS of, interacting the way that it's supposed to, making its memory, regulatory, and plasma cells. Plasma cells, the first antibody set that they're going to make are IgM. We're going to talk about what that is, but that's the snowflake-looking guys. If that looks like to you, we have a whole bunch of antibodies that are stuck together by their little butts or handles. That's exactly what that is. They're pentamers of it. All right, so back to the T cells. It's directed by the antigen presenting cells on how our T helper cells are going to be skewing that TH1 versus TH2 response, but you're always going to have both. That's very important to remember. T cells always require some kind of MHC recognition. With the B cells, once they're activated, they don't need to recognize any more molecule. They'll just crank out the antibody. The T cells, anytime they interact with, especially the cytotoxic T cells, they have to see MHC1 with something. That's always how it's going to be. So we always have to have MHC recognition. We already talked about this, so I'm not going to go into it again. But T helper cells, ultimately, depending on if they're TH1 or TH2, we have regulation of B cells, of course, T cells, of course, and then macrophages as well. So we can send them back to the front line and have them keep going on and doing their jobs. So that's all going to be dictated by our cytokines. Of course, we have regulatory BS. That's just to keep everything in check. B cells do it and T cells do it. Any kind of T cells will do it. Cytotoxic T cells are going to kill other cells. We already talked about that. They're only going to react to MHC1 with antigen or MHC that they see as having antigen when it looks weird to them. That's only going to be present on our nucleated cells, and it's going to be recognizing our intracellular antigens. And it's going to kill infected and abnormal self cells. We talked about perforins and granzymes and the apoptosis. Again, this coming back to that help to the CD8 T cells, the cytotoxic T cell. That's what I'm trying to say. Again, that's always the antigen came from inside the cell. The call came from inside of the cell. Yeah, I mean, it's false coloring, but yes, that's going to be the situation going on. So these are T cells that are binding on the outside. These red things are the T cells. Binding on the outside of that yellow thing is a cell that they're killing. They can kill other non-self cells as well that way. So our immunoglobulin antibody crap is going to be our, we have two segments. Our variable region is also known as FAB. Hey, variable has AB in it. And then the constant region is also known as FC, C for constant. Just think of it that way, okay? It has to do with how they crystallized them and made the shapes of them or whatever. So that FAB, that variable region, binds the antigen, and the constant region is going to determine the function of the antibody. Gosh, I don't know how you always take longer and longer every time I lecture about this. Like last one time I was perfectly on time. Anyways, so antibody functions. They're going to coat. So it's literally going to stop them from doing their functions. They're going to go help with opsonization, giving handles onto the outside of those capsules so we can grab onto them easier, helping phagocytosis. Neutralization, blocking the spikes and how they can get into cells. Agglutination, causing them to stick to one another. The complement pathway, remember that's going to be the classical complement pathway if we had to deal with antibody. And then we can also bind to antitoxins themselves, not dealing with the cells or anything. That's what your tetanus one does. So we've already kind of mentioned opsonization. Again, it's essentially creating a handle so that phagocytosis happens more easily. Here are classes of immunoglobulins or antibodies. IgG, IgA, IgM, IgD, IgE. There's 5. The pentamer, the pretty snowflake looking one, is IgM. It's always the first one made, always. The next biggest, most important one by far is going to be IgG. IgG is our big guns. Think of it as IgG guns, okay? This is our big daddy. Like this is doing like most of the heavy lifting for your immune system, but it isn't made first. We have to go through the IgM process and then we can switch to IgG later on, which is why it can sometimes take a while for your immunity to really be effective. The idea is if I can expose you via vaccine to something, you will already have made your IgG. And then when we kick you back, show you back to that thing that you had your vaccine to, you can just kick out these IgGs. You don't have to go through the IgM process again. So that's the goal with, vaccines. But IgA is associated with your mucosa. IgM, because it is at pentamer, obviously wouldn't be able to do as many of these things like opsonization and whatever, because it's got this weird ass shape that is just not conducive to it being able to function as well. So that's why IgG is the bigger, more important one. IgD is just the receptor that's on that surface of our B cells. And IgE is involved with allergies and parasites. So we'll talk about that in chapter 17. A titer is a term for measuring the concentration of an antibody in your system and using that as a way to monitor your recent exposure to whatever it is. We know that your first exposure, it takes a while to build up your immunity. You've got to find the right cell that recognizes the fidget spinner. Once you find that antigen presenting cell that can digest it and go present it around, now we have to interact with the correct T cell captain that can help spread around. Hey, everybody activate up. It's time to do our business. All of these cells have to proliferate and differentiate before they can even do any work. And then your antibodies have to switch classes to IgG. So you can see why your immune system takes a while to kick in sometimes. Sometimes it's longer than others. That time between when you're infected and when your antibody response essentially comes around is called your latent period. So obviously when you see something the second time, that's the whole point of vaccination, is that you don't have a latent period. We've already gone through it the first time because you don't have a latent period in your secondary or amnesiatic response. This is a chart showing your levels of antibodies compared. You see my little sad face versus our little happy face. Waiting for those antibodies. So then we have active versus passive, and then natural versus artificial. So active is whenever you're making your own antibodies against something. If you're exposed to something for whatever reason or whatever way, and you're making your own antibodies with memory and things like this, that is active. If it's passive, that means we're giving you antibodies. You are not going to create memory, and it's only going to last as long as the antibodies are around in your body. So if we have natural active, that's you getting sick, like with the chicken pox or something, and then now you're immune to it because you got sick with it naturally. Artificial active is introducing that via medicine. So artificial active is getting a vaccine. All right, natural passive is like when you have a baby and the baby's growing inside of you and the mother's immune system is protecting it, or after the baby's born, when the baby is ingesting breast milk with antibodies in it, particularly in the colostrum, you have quite a heavy concentration of antibodies that are providing immunity to the baby. That's natural passive, right? The baby's not building its own immunity that way. It's just getting it from the mom. And then, of course, artificial passive is when anytime you get any sort of antibody infused into your body for like gamma globulins, we've heard about these things, but it also includes things like Humira and Relenza and things like that, that are anything MAB, like adalimumab, which is what Humira is. MAB means monoclonal antibody. So anything that you hear MAB at the end of, hey, you just learned a thing. That's just antibodies being infused into somebody. You can do it by shot or you can do it by infusion, but that is artificial passive. Cool. Cool. We did it. Vaccination, we already talked about that. The whole idea is to elicit that first initial primary response so we can get that secondary. You can have live attenuated organisms. That's not as common, but some things just you get a better response from the live attenuated. They are technically alive, but they shouldn't be causing disease. They've been manipulated to not cause disease. Most of the time, your vaccines and stuff are going to be whole killed. They're dead, like active, like they cannot reproduce. So there's that as well. And we can also use viral vectors where we hollow out a virus and put in genes of COVID, let's say, into adenovirus, which is a cold virus, and put it in your body so that your body can respond to it as though it were COVID without being exposed to COVID. So we know the flu vaccine is dead. An attenuated example is MMR. And then we have vector, that's Johnson & Johnson. Next, we have these structures that are not the whole organism, but just parts of the organism or whatever. We could have surface structures. We could have vesicles that contain mRNA, like we talked about with the other vaccines for COVID, or we could have subunits of things that you're going to react to conjugated with something else to make a stronger response. So Hep B is a surface antigen. The Pfizer and Moderna COVID vaccines were the mRNA in the vesicle. Get that into your body, you make the mRNA products and react to that. And that's the one that the guy that I studied, the guy that got the Nobel Prize. And then the conjugated ones where we're actually like binding the stuff to something else to make it react better. Booster shots are just making sure that your response is going to last a lifetime. So some of these things don't last a lifetime. You have to keep stimulating your cells to keep them around. And so that's what booster shots are for. Some things will last a lifetime and you don't need a booster shot. Now we already talked about this. We're trying to find better things. I'm not going to test you over this stuff, so cool. And then we have subcutaneous vaccines. This is under the skin. You want to get under the whole dermis when you're doing that, but you don't want to get into the muscle if you're subcutaneous. If you're intramuscular, you do want to get in the muscle. And then you also kind of intradermal. We don't see that a lot, but that was like the case with smallpox vaccines, where they would like scratch the surface and try to get a scar to form. And if it didn't form a scar, it didn't take. So we don't see a whole ton of that anymore, but yeah. And then oral and nasal vaccines, we don't see a ton of those either. If you're allergic to the flu vaccine, which you can be allergic to the eggs in it, then they can use the nasal one that does not have egg in it. And that is a live attenuated version. And also polio vaccine is live attenuated and they give it intranasally. It can revert back to wild type. It's extremely rare, but there's been a case of that happening. So our adjuvants help accentuate immune response to make sure that you're responding strongly. So it's additives that we have into the vaccine to help with that. Yeah, just because feel like you have the flu, just because you feel like you're sore or have a fever or something after a vaccine doesn't mean you're sick. Well, not sick. You don't have the vaccine. You don't have the illness. It's not breeding in you. What you have is your body responding to that vaccine. 99.9% of illness that you will feel in your life is caused by your immune system responding, not by the thing that's making you sick. So if you're getting a vaccine and you're having what you think are symptoms of illness, that's just your immune system responding to the vaccine. That's a good thing. You want your immune system to be responding to the vaccine. So keep that in mind. However, just if you don't have the response either, that doesn't mean you're not responding. It just means that yours is maybe more effective than other people's, just saying. So that is part of the misinformation thing. You can't get the flu from the flu vaccine. It's usually just people responding to the vaccine. That's really what's happening. It's dead. It literally cannot be able to make the flu. It can't. So if you got the shot, unless you got the nasal thing, which would still be really unlikely. But yeah, it's dead if you got the shot. Okay, it's just your body creating a response. Also, if we do even go into this, oh my gosh, mercury in the measles vaccine causing autism was never real. That was not ever a real thing. Okay, just to be clear, so if anybody asks about it, so you guys know and you've been educated about this, There was not ever even mercury in the measles vaccines. Those were other vaccines, yes, that had mercury in them. Other vaccines that had mercury in them have mercury at lower levels than what you get in like your daily diet, like literally. And so yeah, there might have been in other vaccines, but not in the MMR vaccine. The MMR vaccine also has never been linked with autism in any appreciable study ever. Even the first one that was published that people try to quote a lot. They say, oh, this doctor, though, he did this research and he published it. Yep, he has admitted that he falsified the data. Literally openly admitted that he falsified the data. People need to shut the F up about it now. So I'm sick of people saying over the vaccine causing autism when that was never, ever actually related. So anybody that wants to argue about that, come at me, because it's literally been, the guy was tried and like had to redact all of his research and everything. It was lies, actual lies. But because one celebrity, he handled it and like wanted to get into it now, whatever. Anyways, there's questions here. If you guys do want to save the questions for yourselves, I'm not going to go over these answers. But yeah, if you guys did want to take a picture for study purposes later on, they are here. And you guys can always ask me if you can't figure out what the answers, but they're pretty straightforward questions. Does anybody have any questions before we go up to the lab? It's a short day in lab. Yes. You're fine.
The autism thing, isn't that more like linked kind of like just to your brain?
Yeah, yes. It has never been linked to vaccines. Yeah, autism, it seems to be more of like, just like a lot of mental illnesses, right? It's supposed to be like environmental and probably genetic.
Like, no, because the brain is like such like a protected system.
Yes, it is. It is.
How is there any vaccine that could reach the brain?
The only times that we have vaccines that could be affecting the nervous system are ones that would lead to like Guillain-Barré or something like that. So it would be a reaction to the overall like nerves themselves. Again, they're really uncommon. It does exist though, but yeah, but we don't see anything like mental illness or autism being linked to vaccines.
A lot of things can happen, like when you're, you know,
Correlations are common, but not causations. Yes, I get what you're saying.
Say why he lied about that? Like, what was his purpose?
I think he just wanted fame.
Attention?
Yeah. I think that's like the research he was trying to get into and trying to prove. And then when it didn't work out, that happened once when I was working at the CDC, not at the CDC, but with the CDC. They had somebody who was like in their lab that was falsifying their data so that they could get like graduate and things like that. So she was just like literally like doctoring her images. Yes, at the CDC.
That affects like the whole world.
Yeah, yes. And so they had to like go through this whole process of like redacting everything that she had published and like,
For that.
yeah, I don't know what happened to her after that. I didn't really follow that part of it, but
That like affects a lot of people's choices, you know, what they read on Netflix.
yes. Yes. Okay, what was I doing? This. Okay.